Welcome to Brain Ponderings, your window into the wonderful world of brain research. And my guest today is Professor Roshan Cools. She's a professor of cognitive neuropsychiatry at the Donders Institute for Brain Cognition and Behavior and the Center for Cognitive Neuroimaging at Radbout University in Nijmegen, the Netherlands. Did I pronounce it right?
That's right.
Okay,
Nijmegen.
good. I
Almost.
got, I got it. It's a tough one. And she spent her career trying to understand how the brain keeps focused on what it's doing in kind of a continuous stream, cognitive control. I read one of her review articles recently, and early on, she kind of challenged me to read the article completely through without checking my email or uh, other things, which is actually it's a really good point. It seems to be getting increasingly hard to stay focused and but this is really important for making good decisions, and that's what the human brain, well, it evolved to do well. I don't know if it's doing it so well in some cases nowadays. So welcome, Rasha.
Nice to be here.
Okay, so you're from the Netherlands. Did you skate on the canals when you grew up in the winter?
Well, I guess the ice was thawing already
Oh,
when I grew
really?
up, but we did skate on the local pond, maybe every third year or so.
Yeah.
Now it
Oh,
comes in
in the
8 years.
Olympics, I always the Netherlands speed skaters are always the best.
Yeah.
Yeah. Okay, so how did you get interested in science and the brain, and kind of what was your trajectory from your early life to where you are now in maybe 2 minutes?
Yeah, well, I have always been interested really from when I was very small in how people behave and think. I was certainly infected by my by my dad, I guess, who was a pharmacologist,
Uh-huh.
neuropharmacologist. But I think one particular event when one of my cousins developed schizophrenia that really, I think, contributed to me entering this particular field. Um, I think I must have been about 17 or so, or 18, 19, something, yeah, 17. So already
And
studying
he was
psychology,
about,
already
your
studying
cousin
psychology.
was about the same age then?
Yeah, he was he was a couple, 1 year younger, or is 1 year younger.
Yeah.
And I think I had already started studying psychology, but that did push me towards this direction of psychiatry and dopamine. And of course, this was a topic on the dinner table already um, at home.
Yeah. And where did you go to college then to kind of get your basic science background?
In Groningen, University of Groningen, north of Holland, did experimental psychology and neuropsychology. And cognitive scientists and early neuroscientists like Ritske de Jong and Bert Mulder, they actually encouraged me to check out Trevor Robbins' lab, whose group I joined after my undergrad degree in Cambridge to do a PhD. And I guess Trevor is my real academic father.
Yeah,
Um,
I had Trevor on this podcast. It's probably been, well, I don't know, I don't have any track of time as I'm getting older, but maybe 2 years ago, he was talking about obsessive compulsive disorder
right.
on the podcast. Yeah, a made a lot of major contributions, and I'm sure he's a good mentor. You got a
Yeah,
lot of, you were very productive during your time there.
yeah, that was a great time. I have everything to thank to him, really. Yeah, I very much enjoyed working with him. Quite a genius, and we're still in touch. Not working actively together, but yeah,
And then you were I guess your PhD work was a lot related to Parkinson's disease.
that's right. That's right. So I came to Trevor to, with an interest in cognitive control and cognitive flexibility already, and dopamine's role in cognitive flexibility um, and Parkinson's disease seemed to be like I guess a great model to study that, right? So it's known for its motor control problems, but actually it's accompanied by quite significant cognitive control problems as well, right? If we consider the mental rigidity, the problems with multitasking that are present already early on in the disease. So uh, that was the focus of my PhD, and effects of medication, dopaminergic medication in particular.
And they have often people with Parkinson's have impulse control and they may start gambling. It would be interesting to see if my son briefly, this son did some online sports gambling, which is legal now in the United States. If you watch sporting events like whatever, ESPN, they will have advertisements over and over for sports gambling. So it'll be interesting to see if Parkinson's patients more and more get involved with that. I don't know, maybe not.
Yeah, I mean, yeah, that's right, right? It got quite a considerable um, proportion of people with Parkinson's disease develop these impulse control problems like gambling. Not just gambling, some people exhibit other forms of impulse control problems like punding, like compulsive hobbying, or hypersexuality, or some people even become addicted to their medication. And so that really illustrates the kind of confusing relationship between dopamine and cognitive or impulse control, right? So on the one hand, dopamine is a key ingredient for cognitive enhancing drugs
Yeah.
like dopa in Parkinson's disease. It's also, well known to improve various cognitive functions, like mental rigidity is really released by the medication. But at the same time, dopamine can also contribute to these types of behaviors like pathological gambling. So that was really the starting point actually of my PhD. How can it be that the same neuromodulator has such different effects, not only in different people, but also within the same person, depending on what kind of behavior you look at?
Yeah. And okay, so can you kind of just def define cognitive control and give some examples?
Yeah.
It's
So
kind of obvious, but it might be helpful for people to understand from their daily lives what they're
that's
doing. Yeah.
Yeah, so I guess cognitive control is a really ill-defined phenomenon. So, and it's also, I think the definition is also evolving, but classically it refers to the sort of that set of mental processes that help us obtain our goals by resisting distractions, by resisting, temptations, by restraint and suppressing our instincts and our habits when they are inappropriate for the current context. So it's kind of this kind of phenomenon is studied classically with Stroop tasks or Erickson-Flenker tasks.
Wow.
When exercise is good, or I don't know, not eating red meat is good for the world, you might have set yourself that goal, but can you actually, pursue that goal, select the actions that are in line with that higher order goal, or do you go for the easy fast food, or sleeping in, which is more attractive on the short term? So I guess that's a real life example of cognitive
Yeah,
control.
I was thinking about this uh, from, I was thinking back to when I was undergraduate in college and going to classes and, sitting through lectures and um, kind of daydreaming, or your mind kind of wanders off the focus of focusing on the lecture. And then, we have a textbook to kind of fall back on. Um, and so you're kind of weighing, and your brain is weighing the probability of, okay, is it okay to let my mind drift off a little bit because I have the textbook to fall back on, and I can do that later. And then when I was teaching, I taught neuroanatomy for medical students when I was a professor at the University of Kentucky, and we're just starting. Uh, no, we actually didn't have PowerPoint then, but we would give the students copies of the notes. We're actually using overhead transparencies where you draw on the light and it projects it on the screen. So we would give them the notes, hard copy, and it was interesting. Some students didn't even show up for the lectures because they had the notes. Um,
That's
but
right.
anyway, so
That's
okay,
my constant struggle now, right? So we
yeah,
can record our lectures and no one actually turns up, so you end up speaking to an empty college room. Yeah,
yeah. Or they
cool.
get now it's a PowerPoint they get, right, of
Yeah,
the presentation.
but that's right. I mean, so what you're pointing out is that cognitive control is recruited, particularly when you have to maintain in your mind's eye what's relevant, rather than sort of in your centric, well, when you can't actually perceive or perceive the relevant thing in front of you, you have to keep it in your working memory,
Yeah.
if you will. You have to keep the relevant goal representations in your working memory. That's often associated with cognitive control. That's right.
Okay, so what brain circuits are involved in cognitive control? What brain circuits have you been studying?
Yeah. So, of course, again, classically, this type of restraint or ability to resist distractions, to resist mind wandering, temptations, that's kind of everyone associates that with the prefrontal cortex. It's not one thing, it's not a unitary thing, the prefrontal cortex, but I think it's fair to say that in general, cognitive controls implicated the prefrontal cortex. But as I sort of implied earlier, I think this construct of cognitive control is also evolving a bit, in the sense that we increasingly recognize that in order to adapt flexibly to our environment, adaptive cognitive control is not just about pressing some button to kind of turn on, working memory, or to turn on um, the goals in your head. It's also very much about learning what actions, what tasks, and what states in the environment are valuable. And in that sense, all the circuits important for learning, based on experience across multiple time skills are
Hmm.
important for
Yeah.
cognitive control. And in that sense, you could argue cognitive control really involves recurrent circuitry across the entire brain, um, involving interactions between different parts of cortex and many different subcortical structures. Of course, much of the literature is about that interaction between the prefrontal cortex and the basal ganglia, different corticostriatal circuits. And I
Now,
get to talk about that.
when I first learned about the basal ganglia, the striatum, uh, humans, the uh, putamen and the globus pallidus, I learned about, from the control of body movements, and of course these neurons are affected in Parkinson's because there's loss of dopaminergic neurons that control them. But then we also learned about its importance in like uh, habitual behaviors, like repeating the same thing over and over. Like if you have a job where it's kind of repetitive, you're doing a sequence of movements or whatever. that repeat and repeat, your mind can kind of drift off and you can actually be thinking about something else while you're still effectively doing the task. And so that, I think my understanding, that was kind of like one of the early functions of the basal ganglia in behavior rather than just controlling body movements on a sub-second basis. So,
Yeah,
yeah.
exactly. That's right. So maybe at some point we thought basal ganglia is important for movement. Then we started to realize it's important for learning these stimulus response links and automatizing them, procedural memory, the kind of learning and memory involved when we learn to ride a bike. You know, we don't have really explicit representations or declarative memories of, you know, the type of knowledge we gain when we learn to ride a bike, but it's these stimulus response habits. But I think today we recognize that the basal ganglia, maybe not the same subcomponent structures of the basal ganglia, but that other subregions of the basal ganglia are also important for cognitive control. So for behaviors that are not already automatized or that require us to keep goals in our minds, you know, that require us to direct actions at our goals. So I think the basal ganglia is important for motor control. It is important for habit formation, but it's also important for directing actions at current goals. And it is important for instinctual responses driven by biologically significant events for motivation. So there's these multiple corticostriatal circuits, and I bet Trevor talked about that 2 years ago as well. These multiple segregated corticostriatal circuits that are involved in motivation, cognition, and action.
He talked about it some, but actually not in a lot of detail. And so, right, so let's get into the actual circuits. So a lot of its work, as is often the case, or usually the case, and the kinds of things you and I are doing, a lot of the early evidence came from animal studies, or at least the evidence getting down to what neurons are doing what, or what brain region or part of a brain region, because you can, in rats or mice, it's okay, you can get approval by the Animal Care and Use Committee to do, selectively damage part of a brain of an animal and see how it affects their behavior. You can actually inject. One thing we did a lot was like seizure model of epilepsy where you inject an excitotoxin, a chemical that excites the neurons to death and kill neurons in a certain brain region. If you do it in the hippocampus, they have impaired learning and memory. If you do it in the striatum with another toxin, actually they have like Huntington's disease, like because you're completely wiping out the. But talk about the circuitry, all the circuitry here between the striatum and the prefrontal cortex. Uh, what neurotransmitter uh, are involved, and kind of the core circuitry of glutamatergic, GABAergic, and then how they're modified. That kind of core circuitry is modified by other neurotransmitters.
Yeah. So,
That's a lot.
well, I mean, so we've just talked about this frontal cortex, right? And this, which is kind of connected strongly with different basal ganglia pathways, which then kind of through the globus pallidus and the thalamus projects back to the cortex again. And there's evidence for these circuits to be kind of topographically selective. So let's say parts of the frontal cortex that have been implicated in motor control are most strongly connected with parts of the basal ganglia that are particularly implicated in motor control. And then we talk about premotor cortex being connected most strongly with the putamen, for example. Those are structures implicated in the controlled movement. And then in addition to that, a sort of separate circuit, and it's not completely separate, of course, there's important interactions between these different parallel circuits, but for the sake of the argument here, we also have parts of the frontal cortex that are rather implicated in working memory or cognitive control. Then we talk about the dorsolateral prefrontal cortex, for example, which is much more strongly connected with the cognitive striatum, if you want, cognitive parts of the basal ganglia. So that's the caudate nucleus. And then we have parts of the frontal cortex, let's say the orbitofrontal cortex and the medial frontal cortex, that are implicated in motivation of behavior. And those are most strongly connected with the ventromedial parts of the basal ganglia or striatum, which is the accumbens, for example. And so we have these different parallel circuits that talk with each other.
And that's all glutamatergic neurons, excitatory glutamatergic neurons.
Well, the projection from frontal cortex to those neurons in the, to the input nucleus of the basal ganglia, the striatum, is glutamatergic. But then the striatum, the median spiny neurons in the striatum, then communicate with the thalamus, which is this relay station, which relays information from the striatum back to cortex, is very complicated. So there's inhibitory, GABAergic connections, and there can be multiple of them. In fact, we have these direct and indirect pathways with different neurons in this striatum connecting with the thalamus through double or triple inhibitory pathways. So it's these through those GABAergic connections, the striatum, this input nucleus of the basal ganglia, is in a position to either release activity in the thalamus or suppress it. And this is, I guess, the basis of these so-called direct and indirect pathways of the basal ganglia, which together form this kind of perfect neuroanatomical arrangement for selecting relevant cortical programs while at the same time suppressing irrelevant cortical programs.
Okay, and then now talk about dopamine, which I guess is your favorite neurotransmitter, at least the one you're working on the most.
That's right. So activity in these cortico-frontostriatal circuits is I guess controlled by glutamate and GABA, as we just said. But what dopamine then does, that originates from these neurons in the midbrain, ventral tegmental area, and the substantia nigra. That's in the brainstem. They actually project to quite widespread regions of the brain, mostly the front part, so the frontal cortex, as well as the basal ganglia. But in the basal ganglia, they act on receptors, dopamine receptors, that are located on these median spiny neurons in the striatum. And there's actually two different types of dopamine receptors. So there's the D1 receptor family that's located primarily on the neurons that are part of the direct basal ganglia pathway. Activity in this direct pathway of the basal ganglia is associated with the amplification, if you want, of task-relevant cortical programs. If dopamine acts on those D1 receptors that are mostly localized in this direct basal ganglia pathway, activity is further released. By contrast, there's a whole different type of family, D2 receptors that are localized on the striatal neurons that are part of the indirect pathway. And this activity in this indirect pathway is actually implicated in the suppression of irrelevant cortical programs, if you want. And if we look at what dopamine does to these D2 receptors, is actually inhibitory. So it suppresses the inhibitory effects of this no-go pathway. So through acting on these D1 go pathway neurons or direct pathway neurons or these D2 no-go pathway neurons, dopamine can have this interesting impact on kind of task relevant versus task irrelevant activity. It can promote the amplification of task relevant cortical programs and at the same time reduce the impact of task irrelevant, reduce the inhibitory impact of task relevant programs.
Okay, so there's these dopamine neurons in the substantia nigra in the ventral tegmental area. Their axons project to these medium spiny neurons in the striatum. And I guess the different medium spiny neurons or maybe different parts of the dendrites of medium spiny neurons have more or less of D1 or D2 subtypes of receptors. And so whether or not dopamine is increasing or decreasing their activity kind of depends on the existing structure. Am I right on that? The structure and where the receptors are essentially, and where the axons have formed synapses.
That's exactly right. So there's different types of receptors that are located on different types of neurons, the activation of which also has different consequences for activity in cortex.
Right. So you have this kind of set structure, and the things we're talking about are happening quickly, right? They're happening with you and I right now, with everybody. And so the structure is kind of set on that time scale. It's structured, although there may be some subtle structural changes with long-term memory or so on. This is happening rapidly, so it has to be, rapid changes in the neurotransmitter signaling. And one thing that, so I was always interested in what we call second messengers. That is these molecules that when neurotransmitter binds to a receptor, some molecule increases or decreases in the cell. So in the case of the dopamine receptors, they either increase or decrease cyclic AMP levels, which is a second messenger. And I guess I presume that cyclic AMP is then meeting either the activation or inhibition of the neurons which dopamine's being released on. Is that right?
That's right. I mean, so I must say that, I'm not a molecular biologist, right? So the type of studies we do is really at the cognitive level and what happens when you manipulate dopamine systemically. So the kind of temporal precision that you're talking about is often lost in our studies when we look at changing dopamine levels with a drug, for example, in Parkinson's disease or in healthy volunteers. But there's reason to think that when you change dopamine levels at that sort of, tonically, let's say, it'll impact these more temporally precise signaling. So it'll impact the functions also associated with this kind of fast timescale changes.
I mean, I was kind of a friend with Paul Gringard, who got the Nobel Prize for, he's essentially looking at this protein phosphorylation. So this cyclic AMP I mentioned, it can affect the function very rapidly of many different proteins, ion channels in the membrane, or whatever. It can even affect gene expression. So there's some gap between, like, in knowledge. There's a knowledge gap, and this is true and essentially all over neuroscience. There's a big knowledge gap between what's going on at the molecular and cellular levels and behavior, and it's really hard to bridge that gap.
Yeah,
Yeah.
that's exactly right. I mean, that level of analysis that you're talking about is even a step further from the problem that we're facing
Yeah.
based on
Yeah.
bridging the neurophysiology and the
Yeah.
cognitive neuroscience, right? Where we have drugs that change, in this case, dopamine for hours, and the neurophysiologists measuring changes in neuronal firing rates or dopamine release across, yeah, minutes or seconds.
Okay, let's get into your work. So you work on humans, and you have to be able to, um, yeah, you you develop, I guess, behavioral tasks that can allow you to answer some piece of the puzzle you're trying to solve, and then you you have to be able to, or you have measured dopamine levels, at least at kind of a brain regional level, or well, focusing on striatum, I guess. And then, yeah, and you've also been doing functional MRI. So take some time and talk about how you develop these approaches and the new things you've added to what was already known. So essentially, that's the rest of this podcast. But
Yeah. So I guess maybe we can go back to the sort of work I started with
okay,
when I was doing my PhD with Trevor. And the starting point there was these contrasting effects of dopaminergic medication um with some forms of cognitive dysfunction being remedied by these drugs and others actually being worsened or actually an impairment being induced by the medication. So that was this kind of paradox that we started from, and we we I guess did work or set up some studies um, to look at what is the origin, the circuit origin or circuit, the system level origin of those contrasting effects. And one factor that's relevant there is the, I guess, well-known inverted U-shaped relationship between dopamine and cognitive function, that not only too little, but also too much dopamine is bad for cognitive performance. So, we need a finely tuned level of dopamine that is optimized for a particular function. But the other point is that dopamine acts on these, on multiple distinct corticostriatal circuits. So not just the motor circuit, not just the cognitive circuit, but also the motivational circuit. And so the idea we wanted to test there was that perhaps the dopamine medication doses that are necessary to remedy the movement problems associated with the disease are actually too high for corticostriatal circuits that are relatively unaffected in Parkinson's disease. So I guess it's relevant to clarify that in PD, there's severe dopamine depletion in the striatum, but it's not uniformly distributed. So the depletion is most severe in the dorsal frontostriatal circuits, whereas the ventral frontostriatal circuits implicated in reward learning and motivation, they're relatively intact. So the idea was that if we give dopaminergic medication to people with Parkinson's disease, we might actually detrimentally overdose this relatively intact ventral frontostriatal circuit. And so to test that, we had people with Parkinson's disease complete a battery of cognitive tasks, some implicating the severely depleted dorsal frontostriatal circuitry and other tasks implicating the relatively intact ventromedial corticostriatal circuitry. And we predicted that these medication doses would have diametrically opposite effects on these sort of dorsal depleted functions and ventral intact, originally intact functions. And this was the
Is
case.
that levodopa?
So we would look at levodopa, which is a precursor of dopamine. And the reason people are given levodopa is because it crosses the blood-brain barrier. Unlike dopamine itself, you can take dopamine, but it doesn't cross the blood-brain barrier. But people also are often given prescribed dopamine receptor agonists, right? Like bromocriptine, for example, or cabergoline. And these can also be taken and they stimulate dopamine receptors. Some act primarily on D3 receptors, which are most abundant in the ventral or limbic striatum, and they are indeed associated with gambling, the impulse control disorders that are seen with medication. There is sometimes seen they're most prominent with these D3 receptor agents. But anyway, you're asking about the kind of work we do. I started to do them with Trevor, but sort of the principle applies also to the work that we do now, because we had in this original study a whole series of tasks that look at cognitive control, but different types of cognitive control. And that's, I think, a key point, that cognitive control is not really one thing. It's multi-computational, implicating different corticostriatal circuits. And it's only the type of cognitive function associated with the dorsal frontostriatal circuitry that was actually improved by dopamine, whereas the type of cognitive function, in our case, we looked at things like reversal learning, reward-based reversal learning, or punishment-based reversal learning. That type of function was actually impaired by dopaminergic medication in Parkinson's disease. Now, of course, the other thing we can do is measure blood signals while people perform these kind of tasks using MRI, and then look at which circuit is modified when people take medication compared with when they take either placebo or when they don't take medication. And what we can see is that these detrimental effects of the medication in Parkinson's disease are indeed accompanied by modulation by these medications of circuits, frontostriatal circuits, that are relatively intact. So those are the ventromedial frontostriatal circuits. Whereas the beneficial effects of this same medication in Parkinson's disease are accompanied by modulation of the more dorsal frontostriatal circuits. So that's just one illustration of the type of work we do. I guess a really question that, yeah, so maybe I'll leave it to that. Yeah.
Okay, and so one thing that you did, among many things, important findings, was that, so all of us have some kind of, if you will, basal level of dopamine tone when it comes to cognitive control. And so a drug that increases dopamine levels at the synapse or activates a certain dopamine receptor when given at the same concentration to you and me, we may have somewhat different effects on our cognitive control. And you haven't mentioned attention deficit hyperactivity disorder, ADHD, right? Right, but which, I mentioned the daydreaming and stuff. So, right, I mean, someone with ADHD, can't keep focused on things very well. Their brain keeps jumping between different thoughts, I guess, or trains of thought. So talk about how you showed that kind of there's different basal levels of dopamine that are related to yeah the, the effects of a drug like methylphenidate, which is called Ritalin, right? That in at least the United States, that was the trade name originally.
Yeah, yeah,
It,
Ritalin or Concerta, which is the um, slow release version of Ritalin. But
yeah,
yeah, Ritalin or Concerta, they're both the active ingredient is methylphenidate, which is this dopamine transporter blocker. So it blocks the reuptake of dopamine after it's been released in the synapse. So it basically prolongs dopamine, the action of dopamine after the brain has released it, you know. Yeah, so um, I guess after my PhD, I went to work with Mark Desposito at UC
oh,
Berkeley for postdoc. And so with Trevor, I'd focused on the basal ganglia and cognitive flexibility and, Parkinson's disease. But then I wanted to learn much more about fMRI and pharmacological fMRI in particular, and decided to approach Mark, who had already focused a lot on dopamine's role in the prefrontal cortex. So, and he was really pursuing that idea that's grounded in a seminal work by Pat Goldman-Keat,
oh, yeah.
that neuromodulators like dopamine in prefrontal cortex, they actually stabilize and sharpen or increase the precision of the representations of our goals in working memory. So they sharpen working memory representations. And that's how dopamine would enhance cognitive control. So that was a kind of a different perspective on this problem of cognitive control and dopamine, right? And it's this mechanism that had been implicated in ADHD and Ritalin. We actually still think that Ritalin in part enhances cognition by acting on the prefrontal cortex and by sharpening the goal representation in our mind's eye. We're better able to remember, to stay focused on that goal, and to also protect it from distractions.
And her work was a lot in non-human primates, right?
Yeah, exactly. So of course her early work in the 70s, she did these 6-hydroxydopamine lesions in prefrontal cortex, and then showed this deficit on the delayed response task of working memory that was almost as large as that seen after complete ablation of the prefrontal cortex. That was focused on this principal sulcus, this dorsolateral prefrontal cortex. And yeah, that was really quite revolutionary, because until then, dopamine had mostly been studied in the context of the movement problems
Yeah,
seen
yeah.
in Parkinson's disease. And then her work really taught us dopamine is really much more cognitive, is involved in all this cognitive stuff rather than just movement control. Yeah, that's funny, because of course in the I guess 90s, dopamine became particularly famous because of the reward learning story, Wolfram Schultz's contributions, and which accounted for a lot of the behavioral activation effects, the motivational effects that had been seen in behavioral pharmacology already before. But those lines of work on, kind of reward learning in striatum and prefrontal cortex and working memory, they were completely segregated.
Yeah.
And so the effects of Ritalin, you're asking about Ritalin methylphenidate, right? In ADHD, that sort of question has been studied a lot in the context of dopamine and prefrontal cortex, whereas Parkinson's disease and addiction and gambling and impulse control have been studied in the context of dopamine's role in the basal ganglia. So there's these kind of two separate stories, which I guess the field is now beginning to integrate. Like, it's beginning to ask how might we integrate or synthesize these two relatively separate roles of dopamine in the prefrontal cortex and working memory and goal stabilization, and on the other hand in the basal ganglia in reward learning and cognitive flexibility.
Right. So, but I think somewhere I've seen, I don't know where, maybe some podcast or something, that some people, so there's like some subgroups of people in the techies in Silicon Valley that they play around, kind of do, they call brain hacking, trying to improve their cognitive performance, by whatever, taking low doses of
Yeah,
psychedelic or Ritalin, I think,
Ritalin.
methylphenidate too.
Yeah, or
Um,
modafinil, or yeah, there's a bunch of these smart drugs. Yeah.
and are there, for like you and I, can we enhance our performance anymore? Or
Yeah.
are there dangerous side effects? Because I think that's kind of important.
Okay, so I think this actually we need a lot more research on this, huh? But so I think the jury isn't out actually. So what's definitely true is that the effects of drugs like methylphenidate, like Ritilin, will depend on the baseline tone of dopamine, the amount of dopamine you have in your brain already. Like we can see that in the lab, so we can measure the amount of dopamine that is that you synthesize in your brain. That's like a relatively stable, interestingly stable thing. So if you measure it one day and you measure it in another day, that's highly correlated. And we can see that people who synthesize a lot of whose brain makes a lot of dopamine, they will benefit from methylphenidate much less
Yeah.
than people who synthesize who don't synthesize dopamine as much, right? So baseline level of dopamine determines how much you benefit from the drug, but it's much more complicated than that, because the effect doesn't just depend on how much dopamine you have in your brain already, but it also depends on the task amount.
Uh,
So increased in dopamine might be good for one task, but
uh,
not for another task. So different types of tasks have different optimum levels of dopamine.
hmm,
And the real open question is whether drugs like Ritalin, which increase dopamine, but not just dopamine, and they don't just increase dopamine in the prefrontal cortex, they also increase dopamine in the basal ganglia.
yeah.
Do they um,
Hmm.
do they do they kind of shift the system towards one state at the expense of the other, right? If so, then that's good for one type
Hmm.
of environment, but
Hmm.
it's bad for another type of environment,
Hmm.
right? So, for
Yeah.
example, Ritalin might help you focus, which is good in the classroom when you need to focus on your teacher and you complete your math assignment. But it might be bad on the school ground when you can actually benefit from some distraction and you will become more creative. You can interact with
Yeah.
unexpected, I don't know, social input, the kind of lack of focus that's bad for the classroom could be good on the school ground, on the playground after school.
Yeah, that makes sense. I mean, our brain is complicated, and
Yeah, but
there's
so that's possible. It's possible that that happens, but I don't think the jury is out yet, because I think
uh,
it's also possible, and this is what the hypothesis we're testing right now, that these drugs, because they act on all these systems at the same time, they're actually perfectly suited for helping us adapt to those changing contexts, so that it helps us be focused when the environment requires focus, but it helps us to be flexible when the environment requires flexibility. So if we give in the lab a task that requires focus, yes, then we can see Ritalin improves focus and impairs flexibility. But what we're doing now is developing task procedures where the demands for focus and flexibility change, they fluctuate over time.
Right.
And then we see whether Ritalin helps us be focused when we need to be focused, but to be flexible when we need to be flexible. And the way that would happen in the brain is by acting on the prefrontal cortex when the prefrontal cortex is important, but on the basal ganglia when the basal ganglia is important. It would work by boosting the circuit that's kind of optimally tuned to the environment at that moment of time.
Right. So Ritalin it's preventing, so when dopamine's released, a dopamine neuron fires, and dopamine's released from the presynaptic terminal into the synapse. And then there's these dopamine transporters in the membrane of the presynaptic terminal that their function is to rapidly remove the dopamine so that then that synapse can prepare for another signal. So Ritalin is inhibiting the dopamine transporter, so it's increasing the amount and time of dopamine increase at the synapse. What about, so that's kind of in my view, from a physiological standpoint, that's kind of an abnormal way to do it. What about increasing the amount of dopamine in a neuron? It's produced as our other, we call monoamine transmitters from certain amino acids, in this case, namely tyrosine. So what about this loading up on tyrosine? I saw you've actually been looking at this, or at least done one or two studies with this.
That's right. Yeah, so I mean, tyrosine is also a precursor of dopamine, right? So it's actually the effects in theory could be quite similar to levodopa in the sense that you promote synthesis of dopamine.
Yeah.
So there's kind of more dopamine is made if you want. So there's then more dopamine to be released.
Yeah.
Tyrosine can also have these cognitive enhancing effects by increasing the amount of dopamine that's synthesized, a bit like levodopa.
But it also increases, it should, right, norepinephrine levels?
Right, right. And this is true for levodopa, for tyrosine. It's also true for methylphenidate, right? So this is a whole different question. How to disentangle effects of dopamine and noradrenaline. That's not trivial because dopamine is also a precursor of noradrenaline. In fact, there was a time where we thought that dopamine didn't have a function in itself. It just served, for making noradrenaline. And now that's clear that that's not the case, that it has a unique function, but it's complicated. And to do that, what you really need to do in the lab or in real life is compare effects of drugs like methylphenidate that increase dopamine and noradrenaline with drugs like atomoxetine, which do not act on dopamine, at least not in the striatum. So that comparison, atomoxetine is also used to treat ADHD, is useful for isolating the role of striatal dopamine specifically. Yeah.
Okay, then, right. So I was reading, well, I guess it was in you had a couple review articles I read, one in Neuron and one in, I think, Trends in Cognitive Sciences or something. And I, I think it was there. One of those, you were talking about this multi-component model. You've kind of, I think, talked, but this gets into like, say if the task you're doing is mainly visual, like which a lot of things we do, right, are visual, and reading, whatever. And so this, or maybe I read about it somewhere else. There's this multi-component model where you have like three different loops, the phonological loop, the visuospatial sketch pad. So I guess the idea is like you've got this phonological, so I guess language loop, then visuospatial sketch pad, I guess the idea that you're keeping in your working memory some representation of something, the object or something. And
Mhm.
then there's this central executive.
Yeah, that's like Alan Badley's model of working memory. This, yeah.
And how does that relate to these I mean, it must relate to these circuits you've been talking about, the frontal cortex, prefrontal cortex, and the striatum.
That's
Or
an
not?
interesting question. I haven't written so much myself about
Yeah.
sort of Alan Badley's model of working memory, where indeed this kind of phonological verbal system is distinguished from visual spatial and a central executive. Um, one problem with these kind of models is that they're somewhat homuncular, right? So then the central executive um, requires a sort of separate little man, a homunculus in the prefrontal cortex often. This is often
Uh,
associated with the prefrontal cortex and the
uh,
visuospatial sketchpad and the phonological loop,
yeah.
maybe with different parts in the back of the brain where this prefrontal cortex would then interact with these subcomponent processes. I mean, this is in line with the classic idea of cognitive control, where also people who are good at cognitive control, they're good at exerting restraint because they can press this button. I think we're now kind of changing our minds about what makes a person good at cognitive control. Is not because, their prefrontal cortex or their central executive work so well. It's partly because they've acquired, based on experience across multiple time scales, um, knowledge, or they've learned to assign value to those actions that are most optimally tuned for the environment that they're in at that moment. So according to this sort of newer idea about cognitive control, which really emphasizes that cognitive control is much more about learning and inference, about experience-based, value-based choice, about learning from experience, right? Rather
Yeah.
than
Yeah.
sort of a thing that's to do with capacity. Um, I lost my train of thought
No,
there.
no, but so then what about the early life environment then and how that, for example, is ADHD more common in kids who've had certain type of early life experiences compared to someone? So there are any environmental influences on
Yeah.
this, or is
So
it?
when I talk about experience, I'm talking about experience across multiple time scales. So it's experience like in real time, across seconds, minutes, across
Oh, oh,
the lifespan, across development. But you can also think about experience across even longer time scales, right? So it's clear that ADHD has a genetic component, maybe even an epigenetic component. Um, uh, so saying that it's a function of experience doesn't mean it's nurture versus nature, right? That distinction is not something I'm claiming. I'm just um, I do think there is also a component of childhood experience, and in the extreme version of this account, some forms of what we call mental health problem. Well, I guess some mental health problems could be reconceptualized as reflecting kind of normative adaptation to an environment people have experienced in their childhood. I think I can illustrate this better with depression and learned helplessness. Um, so I guess it's well accepted that some forms of depression are accompanied by learned helplessness, where learned helplessness is a kind of a belief, corresponds to a belief that the world in which you live and with its stressors is uncontrollable. And I think there's a lot of research suggesting that that belief might be formed in early childhood. Early childhood experiences of no control, right? Of having no control over stress can contribute to this learned helplessness behavior in adulthood. And so one open question is whether ADHD or people who tend to, I don't know, have difficulty focusing is associated with experiences earlier on in life with an environment that's very unpredictable or very volatile, very changeable. Because in a changeable environment, it's not that adaptive to focus on one thing all the time. So in an environment that's very changeable or very, very volatile, a sort of overly flexible or distractible mindset might actually be adaptive. So in that sense, unfocused or distractible or flexible behavior could be seen as an adaptation to a world that is perceived as highly volatile. But I think this is clearly not the only factor. There's certainly genetic factors there as well. But of course, it's an interaction between genetic and environment.
One environmental factor that seems to improve my cognitive control a lot is physical exercise. Have there been any studies looking at the effects of physical exercise on these cognitive control systems you've been talking about?
Yeah, I think that's fascinating. Yeah? So isn't it interesting that we all study these effects of these drugs and all kinds of interventions when, you know, the beneficial effects of exercise anecdotally are excruciatingly large, aren't they?
Right. Yeah. I had about 5 years, 6 years ago, I had a bad accident on a mountain bike. I had to have multiple surgeries. I couldn't exercise, and I've been like a distance runner and riding mountain bike, and man, I got really bad depression
Yeah,
because I couldn't exercise really much at all for several months.
yeah,
And then I find just on a daily basis, like you feel good after you exercise,
yeah, exactly.
and I can focus. So
Yeah, no, I agree. I have the same experience. There is I know of there is a lot of research actually happening in my institute. I'm not doing that myself on exercise and the beneficial effects, not just on cognitive functions and mood, but also on Parkinson's disease. So even progression of neurodegenerative disorders is slowed
yeah.
with exercise. So I think the impact of that is currently, at least in folk terms, still rather underestimated. And I think we need a lot more work on that, also on the effects of exercise on the neuromodulatory systems.
Yeah, I mean, that's
Yeah,
another grant you could get funded on easily, I think, just to look at, do the same task, do the PET imaging of dopamine and, randomly assigned to exercise or not, or, I mean, I think that'd be really
yeah.
interesting.
Yeah, well, I mean, so I've become quite interested in stress recently, and stress is this another one of these ill-defined terms, but it really refers to arousal, right? Now, arousal and stress are not unified systems again. And in fact, all the large neuromodulators are released upon stress, upon
Yeah.
arousal. And
Yeah.
exercise is accompanied by increase in arousal, but a particular form of arousal or stress, namely controllable stress, right?
Yeah.
So and so I think there's something really interesting in an exercise to the degree that it increases stress hormones or stress or arousal.
Well, there's an evolutionary basis, right? There has to be what my lab did a lot of work on intermittent fasting and its effects on the brain. And, so I've thought a lot about, getting food is like the major thing organisms have to do to survive, right? So a lot of your brain systems are geared to being your your brain is evolved to function very well in a food deprived state in terms of uh, and you have to typically a lot of animals have to expend a lot of physical exertion when they're in a food deprived state to get food. And so it kind of makes sense that your brain should be functioning really well and highly focused on the goal of getting uh, food. So you're getting physical exercise in a fasted food deprived state, and it seems like that really makes your brain focus.
Yeah. Well, so now you touch on another thing that interests me a lot is a distinction between this goal-directed effortful action and there's another type of effort that's instinctual. So
Wow.
when you anticipate reward, we can so the anticipation of reward is associated with two types of systems, right? One is this kind of cognitive, the system that involves cognitive effort, focus, cognitive control. It's computationally expensive in some sense because it has to be tuned to the environment. But then there's also the instinct of activating behavior and approaching and running in anticipation of reward. And that's not computationally expensive. It's not involving any focus or cognitive effort. It's Pavlovian, it's pre-programmed, right?
Yeah, well,
So
I'm thinking more about, say, a hunter-gatherer ancestors having to, remember where the whatever prey animal is and have like a cognitive map in your brain of the territory that you
yeah,
occupy, or if you're foraging, right, remembering where certain fruit trees are located, what time of year, and that, and kind of
yeah,
planning your day, where you're going to go, and where it's more likely that you're going to acquire food than just kind of randomly going around until you see some food and then going after it.
yeah. But how do we decide, right? Which of these
Yeah.
two? So this foraging, this cognitive mapping, model-based system, how do we prioritize that over this kind of pre-programmed instinct driven system?
And there's some nice work by Passingham and Wise. They had this nice book on prefrontal cortex. Is fairly old now, but they did these kind of develop these ways to assess in non-human primates foraging strategies. How long do you stay in a place where you've been consuming all the berries and there's hardly any left before you decide to go to another place? Um,
Yeah, these
yeah,
explore exploit dilemmas.
yeah, yeah. Okay, really interesting problems. Um, did you, so this reward, the kind of cost benefit issue is kind of the bottom line of this in a lot of ways that we're talking about, what your brain trying to make a decision on whether it's better to do X or keep doing what you're doing. Um, do you ever do the prisoner's dilemma? Um,
Um, I know it. I don't do it. We don't
okay.
use that kind of
Okay.
those kind of procedures, but
That was kind of the economic, I think that's John Nash, right? The mathematician who had schizophrenia, he was involved in like, and from a mathematical level, that making economic decisions on what's cost versus risk type
that's right.
decisions. Yeah. Okay. But that's kind of what you're looking at though with
Yeah, that's right. And not just sort of at the level of actions, but also at the level of, um, I guess, behavioral strategy. So cognitive control is very much about learning which whether this kind of foraging strategy, model based, cognitive mapping strategy is paying off, whether exerting the cognitive effort to adopt that kind of expensive strategy is worth the effort, is
right,
worth it,
right,
is valuable,
right.
or whether we have to instead rely on this kind of cognitively more frugal system that relies on instincts or habits. And that's something we learn over time based on experience,
Okay, yeah.
something we can infer. So dopamine is not just about, boosting our instincts to get the reward or boosting the effort, uh, to engage in this kind of complex foraging behavior, but it's actually in computing which of these strategies is optimal given the environment. And if that's true, then drugs like methylphenidate will help that computation, and they will help us select which of these strategies, the foraging, the complex cognitively effortful foraging strategy, or rather relying on instincts, which
Yeah.
is good, right? On average, instincts are good, right? They've become instincts because they work well.
Yeah, that's true.
But we should not avoid relying on instincts all the time.
Right.
After all, our capacity is limited. But making a choice, arbitrating between these systems, that's the key. And I think drugs, yeah, I get neuromodulators like dopamine, but also the other neuromodulators, they're important for
Yeah.
making that sort of choice between those different strategies.
Yeah. Okay, I think that's a good place to end because it kind of summarized everything in a more global view.
Yeah, I guess there's much more to say, but for now, maybe the next one would be in a couple of years.
Yeah, okay. Yeah, if I'm still doing this podcast in a couple of years, I'll touch base again and see how you're doing.
Mhm. All
Okay,
right.
very nice to meet you. Thanks for taking the time. I know you're busy and keep up the good work. I'll try to keep following it.
Thank you, Mark. It was nice to talk with you.
Okay, goodbye.
Bye.